I now want to introduce Dr. NAMRATA SHARMA, who will be talking to us on corneal ectasia. Dr. SHARMA, as we all know, is the secretary. Wonderful job of the AIOS. Unbelievable amount of work you've been putting in, totally. Dr. SHARMA.
Thank you, Jerry. Thank you for your visit here to India. It's always a pleasure to have you here. And thank you for making me a part of this very important course in genetics. I would be talking to you about genetics of corneal ectasias. And the causes of corneal ectasias could be primary or could be iatrogenic. And genetics does have a role even in iatrogenic cases, because if something is linked genetically, then the propensity for ectasias after various surgery increases in these cases. Keratoconus is the most common form of ectasia that we see, and in all the literature that is available, it is generally tends to be bilateral, although it is asymmetrical. But it can be unilateral, and surprisingly, one study showed that 41% keratoconus patients were unilateral during the diagnosis. We currently have this ABCD classification of keratoconus which we follow. An important thing to know from this diagram is that there are various types of collagens which are present in various layers of the cornea. So type 1 and 5 are present in stroma, 1, 3, 4 and 12 are present in the Bowman's, and 4 and 7 are present in the basement membrane. And these essentially become important when we are looking at genetics of keratoconus. There is reduction of epithelial and stromal keratocytes. If you see, if you look at the molecular level, there's reduction of epithelial and stromal collagen content, degradation of corneal membranes and extracellular matrix, abnormal mitochondrial functions, and even deranged lipid metabolism. So there's upregulation of the degradative enzymes, which is expected, and there's downregulation of the proteinase inhibitors. 4 times increase in the receptors of interleukin-1 occurs, which causes apoptosis of the stromal keratocytes, and there's also microtrauma, which releases the interleukin-1 from the epithelium. So if you look at the etiology of the keratoconus, this summarizes it very well. There is a molecular aspect to it, and there is also a mechanical aspect to it. So molecular hypothesis, according to the molecular hypothesis, there is unknown collagen dystrophy that is there. But when factors like atopy, eye rubbing, etc., and inflammation are coupled with it, then it leads to having keratoconus. And there is purely also a mechanical theory to it, where eye rubbing causes changes at the molecular level due to inflammation, and that causes keratoconus. And this essentially summarizes what I was talking about. So if you look at the genes, there are various genes which are cornea-associated genes, like corneal biomechanics, central corneal thickness, keratoconus genes, corneal dystrophy genes, and the corneal curvature genes, and various ocular syndromes also. And I think some of them become important, like this COL-BA2 gene, which becomes important because these are overlapping genes. So just to show, like various ocular syndromes again are overlapping genes with the keratoconus, and that is why some of the syndromic syndromes would have keratoconus. And keratoconus specific genes with no other ocular disease are as shown, and I think LOX is the one which is most important, and there are others which have also been isolated. Now there are various techniques for mapping these genes, which include the standard linkage analysis, the GWAS, the NGS or the next generation sequencing, copy number variation, and mutational analysis. And this is our study which was done at our center, mitochondrial complex 1 gene analysis in cases of keratoconus. And it did show that there are 18 nucleotide variations which are studied, and 2 novel frame shift mutations were detected in this cohort of 20 keratoconus cases when it was compared with 20 healthy eyes. And this is the picture from the same, where there was deletion of nucleotide G codon change from GGC to GCT, and deletion of nucleotide CA codon change from ATC to ATT. This was another study where we looked at VSX1 gene analysis in cases of keratoconus, and no pathogenic VSX1 mutation was identified in this cohort. But there were 2 alterations of VSX1 gene which are noted, and both variations were non-pathogenic. And other genetic loci such as 13q32 may have been involved in cases of pathogenesis of keratoconus, was something that again we concluded from this study. Then candidate genes which are associated with keratoconus are as shown: the VSX1, the MIR184, the DOC9, SOD1, interleukin-1, COL4A3, 4A4, and 5A1, which are primarily the collagen genes. And this helps you to know the disease mechanism. Once you study these genes, then one can identify the biomarkers and also correlate it with the family history. So the genes which are associated with keratoconus, these are the functions of those candidate genes, and genetic conditions are also associated with keratoconus, and this is a whole list of the conditions which are associated with it. If you look at the inheritance pattern, it could be sporadic or it could be familial keratoconus. 90% of the pedigrees with familial keratoconus display autosomal dominant inheritance with reduced penetrance. And then there have been studies also on the monozygotic twin studies where there's higher rate of concordance, greater evidence for primary genetic causation rather than environmental etiology. And the concordance does support evidence of heredity as a genetic factor in keratoconus. And in cases of discordant for keratoconus reading, it was the other things, that is the environmental factors, which are responsible for IRABing for etiology of keratoconus. Then again, this is a study from Narayan Netralaya where they looked at the inflammatory markers, and they found high levels of inflammatory factors such as TNF-alpha, interleukin-C, and MMP9, and altered corneal epithelial and stromal expression of the specific genes at corneal cone apex were there, which drove actually the focal structural weakness in keratoconus. Now, this is the list of the identified genomic loci in keratoconus through various linkage studies, and loci 5q is being replicated in different studies, so I think this remains constant, although all the other factors are a little variable. And keratoconus susceptible genes were reported in different studies based on the different techniques. So that could be one confounding factor, and this is again a list of the various genes which have been identified with keratoconus. So overview of all the investigated genes in keratoconus is highlighted in this diagram, which was published in a review article, and I think the LOX is the one which is the most important. Now, genetic screening does help in these cases. This was a study of 79 Chinese and 9 Greek families of keratoconus, where 91 patients were detected or diagnosed to have keratoconus, and there were 5 variants in VSX1 and TGF-beta1 genes which were identified in these families. So there is no causative gene known for keratoconus that causes the disease directly, and susceptibility genes have to be identified with about 200 polymorphisms in more than 50 genes. Available genetic information as such now is insufficient for keratoconus, and there are no specific pathways which have actually been confirmed. A word about keratoglobus: nothing much have been done by other ectasias. Recent study reported association of variants of this with TMEM45A with keratoglobus, and with pellucid marginal degeneration also, which is a rare disorder. Hardly any studies are available, and only COL5A1 mRNA expression was significantly lower in keratoglobus and PMD keratocytes, and again LOX mRNA expression was higher in patients with keratoglobus keratocytes. So like I said, we need to still study a lot. There is a lot of variation, lot of variability, and lot of overlaps and interlaps which need to be sorted out as far as the genetics of keratoconus is concerned. Thank you very much for your kind attention.
Well, thank you, Dr. Sharma.